welcome everybody to this MycoTalks, which is an international symposium for medical mycology consisting of 30-minute talks on all aspects of medical mycology. We try to strongly encourage a wide diversity of types of talks from people of different career stage, gender, geography, and area. During the talk, you will be able to send your questions to the two speakers that we have today, and please use the Q&A function to do this. You may be communicated also by the chat function by the moderators, but for the questions, please use the Q&A function. If you want to see this talk again or somebody else to see it, there's a link provided there, and you'll be able to see this on the YouTube channel. And in the next slide, you'll see that today's speakers, and we have Bernhard Hube and Geraldine Butler. And I want to thank also the organizing committee that put together a fabulous program of people throughout the year. And just to whet your appetite for the next one, in the next slide, you'll see that the next one, which you must register for, the next one will take place on the 29th of October. And that will be Sarah Gaffin and Alex Brand. And then on the 19th of October, we're going to have Joe Heitman and Tobias Hole. So that's in store. But today, we're going to start off with the first of our two speakers, who is Bernhard Hube. And many of you will know Bernie. He holds the chair and professor of microbial pathogenicity at the Friedrich Schiller University of Jena in Germany. He's also head of the Department of Microbial Pathogenicity Mechanisms at the Hans Knoll Institute in Jena, in Germany. So Bernie did his PhD a few years ago now in the University of Göttingen, and then his postdoc actually in the University of Aberdeen with me, where we first got to know each other. And then he was at the University of Hamburg, and in the year 2000 became he held his first group leader position as head of mycology at the Robert Koch Institute in Berlin. And then he moved to his current location in Jena, and in 2007 he became head of department in that microbial pathogenicity mechanisms department that you've heard about. So Bernie's had a long and distinguished career, published more than 230 papers dealing predominantly with molecular infection biology of human pathogenic fungi, and in particular of Candida. And of course, relatively recent, he and others collaborated to describe the first peptide toxin of a pathogenic fungus in Candida, candidalysin. So at this stage, I want to hand over to you, Bernie, and to give us your talk, which we're all looking forward enormously to hearing.
Right. Thank you very much, Neil, for the introduction. Of course, it's a pleasure for me to be able to speak at these MycoTalks, and I will talk about my currently favorite topic, candidalysin, the virulence and avirulence functions of candidalysin. But before I do so, I would like to stress that I'm dedicating this talk to Frank Otts, a pioneer and world-leading authority in medical mycology, who sadly died a few months ago. So this talk is for you, Frank. Frank was well known for many things, but probably mostly for the book he wrote, Candida and Candidosis, which is still cited a lot, at least I do. And so this indicates that his favorite species is Candida, or the genus Candida. And I also will speak about Candida and about the most common species of the Candida genus. So that Candida albicans, which is associated with animals and humans, it's an opportunistic pathogen which only can cause disease in susceptible hosts under certain circumstances. It's responsible for the majority of Candida infections, more than 50%. It's polymorphic, as it can grow in different morphological forms, like a yeast form, a pseudo-hypha form, and a true hypha form. And because most of us are actually carrier of this fungus. I call it the most common human pathogenic fungus. So this picture is also adapted from Frank's, which he published in 1994 in ASM News, which shows you the principles of Candida albicans. The Candida albicans switch from commensalism to pathogenicity. So normally Candida albicans is a commensal colonizing at a harmless commensal mucosal surfaces of the oral cavity, the vagina and the gut. But very frequently it can cause mucosal infections, also skin infections, and if the fungus is able to invade through epithelial cells, or if the epithelial layer is damaged by surgery or drug treatment, or if it is spread from medical devices, from biofilm of medical devices, it can cause bloodstream infections and can infect almost all organs, causing life-threatening disseminated infections. So today I will talk about the different stages from commensal to pathogenicity, which is basically adhesion, invasion, and damage. And these stages can be further divided by substages, which is ad hoc adhesion and induced adhesion, induced endocytosis and active penetration, and damage and hyphal elongation and destructive activities. And you can also imagine that from these stages on mucosal surfaces as a commensal, especially in the gut, to invading of tissue, the strategies by which the fungus is actually gaining nutrients has to change. So there are certain nutrient acquisition strategies used by the fungus. And sooner or later, the fungus will face immune cells, and therefore immune evasion strategies are also required. And finally, translocation, as I mentioned, when the fungus can pass through the epithelial layers and enter the bloodstream, that's translocation, and there are also specific activities required for that. Today I will start with invasion of Candida albicans into epithelial cells, and here's two different routes, as I mentioned. One is induced endocytosis and one is active penetration. Induced endocytosis is an entirely host-driven process, so you don't need any fungal activities. In fact, you can use killed. However, they have to be hyphae, because hyphae express ALS3, and ALS3 is the main invasion which actually triggers this induced endocytosis. There's also active penetration, and this, in contrast, is entirely fungus-driven. So you don't need any host activities here. Actually, you can kill the host cell, and the fungus is still able to invade host cells by active penetration. Again, you need hyphae. Depending on the cell type, in this case, we are looking at oral epithelial cells, where we have a minor contribution of induced endocytosis, about 25%, and the dominant invasion route, 75% for oral epithelial cells. But other cell types, for other cell types, it's different. For example, intestinal cells are only invaded by active penetration. And this is similar to the invasion of plant pathogenic fungi or mycets. Like in the case shown here, Magna portagriziae, their special structures are built on the surface of leaves, and this is an aprosorium, the special structure. And there are fungal elements which are pushed into the fungal tissue, and there's another structure here which is called an hostorium, and this is surrounded by an intact extrahostorial membrane, and that is very important for the interaction with the host. And we observed that in the case of Candida albicans, the situation is similar. So here you have induced endocytosis, active penetration. This is the tissue underneath, and if you look at this part of the tissue in more detail, then you can see that this part is actually surrounded by a membrane. And that's more clearly seen in transelectron microscopy, where you can see here the membrane surrounding this invasive hyphae. So that's what we call the invasion pocket. So there's an invasion pocket which is similar to the structures of plant epithelial cells, plant pathogenic fungi. So as I said, during the invasion of the host, the fungus requires nutrients including iron. All pathogenic microbes require iron. And so does Candida albicans. And we observed that during invasion, there's ferritin accumulated on the surface of these invading hyphae. You can see the ferritin molecules shown here accumulated on the surface of this invasive hyphae. And we know now that the receptor of this ferritin binding is actually ALS3, because ALS3 mutants do not show binding of ferritin on their surfaces, the cells. So the idea is that ALS3 is a receptor for ferritin, and then you have acidification, which releases iron from the ferritin molecule, and then you have the reductive uptake system to translocate iron into the host cell and to deliver iron to the host cell. So that iron acquisition via ferritin as one example. Another example of nutrient acquisition is zinc acquisition. Zinc is as important as iron as a nutrient. And here we observe a very similar situation as compared to iron. We do have zinc accumulation on the surface of these invasive hyphae. And here the system is different as compared to iron. Here we have a zincophore. And the zincophore is, in this case, is PRA1, a protein which is released into the host cell. It grabs zinc from the host cell and it returns to the fungal hyphae, and it delivers zinc to ZRT1, which is both a PRA1 receptor and a zinc transporter. So therefore we called it a zincophore in analogy to xerophores from bacteria or other microbes. So two different systems for the acquisition of metals. However, I told you that there's a membrane surrounding the invading hyphae, and at the same time I told you that the accumulation of these molecules, ferritin or zinc, on the surface of this invasive hyphae, how can that be if there's a membrane around this? So how does the fungus actually get access to the host cell cytoplasm? So we would propose that there must be damage induced by the invasive hyphae in this pocket during invasion. And here comes EC1 into play. EC1 is a hyphal association, actually the name for extent of cell elongation. It was one of the first hyphae-associated genes identified in the lab of Bill Fonzi in 1993, and it actually belongs to the core filamentation response genes. And these are only 8 genes which are expressed under almost all conditions when hyphae are produced. And in fact, EC1 is extremely highly expressed during invasion when hyphae are formed. So about 10,000-fold expression of EC1 during invasion. And we could show this with qRT-PCR, but also with GFP reporter. And you can see here that there is strong expression of GFP driven by the EC1 promoter in this context. So EC1 is very strongly expressed. And when we look at as an EC1 mutant, then there was almost no differences as compared to the wild type in terms of hyphal formation and hyphal length. The adhesion properties of these hyphae are almost identical to the wild type, and so is invasion. However, there was almost no damage. So EC1 is clearly required for damage. Please also note that invasion per se does not necessarily mean damage. You have to have additional activities. So then we have had a closer look at the EC1 protein, which is actually a polyprotein. And you can see the polyprotein consists of 8 different peptides, which are divided by lysine-arginine sites, KR sites. And KR sites are well known as KEX2 protease cleavage sites in the ER and the Golgi. So these, we propose that these peptides are processed by the CAX2 protease, and we could actually show that already in 2008 by a recombinant EC1. This was shown by Oliver Bade in Berlin at that time. EC1 is digested by CAX2, and we could confirm this using LCMS, that all these peptides exist, and we know now that that it's not only a digest by CAX2, but it's a sequential digest by CAX2 and CAX1. So that means the CAX1 digests after the K. So all these peptides actually end up with the K and the R is removed by CAX1. So then we questioned what is the function of these peptides and which peptide is responsible for the damage. And we could show by using synthetic peptides, EC1 to EC8, that only peptide number 3, synthetic peptide number 3 per se, is causing damage. So that means peptide 3 is the damaging peptide. And the structure of peptide number 3 is actually very similar to melittin, the B venom. That's a very potent cytolysin. And similar to melittin, EC1 peptide 3 is amphipathic. It has a hydrophobic N terminus, 2 alpha helices, and a hydrophilic C terminus. And these data, which are shown now, are not published. It is data with a nanospot assay, which is a very nice assay to actually show the activity of molecules which can cause damage of membranes. And in this case, there's this nanospot assay where you have an array with little holes in it, and you have dyes in this hole, and you can cover this with a membrane. And if you then add EC13 or other components, or in this case this peptide, then EC1 produces pores, and these pores release the dyes from this nanospot assay. That can be nicely quantified. That has been done by the group of Thomas Gutzmann, and he actually could show that depending on the structure of the membrane, whether it contains phosphatidylserine or not, you have very, very strong damage, and it's actually show the evidence that EC1 is a pore producing peptide. So with other words, we have discovered the first Candida albicans toxin, which we have called candidalysin, the missing link between hyphae formation and host cell damage. So coming back to the pocket, candidalysin at the pocket, remember we proposed that the damage would be here at the tip. You could actually show with an antibody, now we have an antibody which binds to a candidalysin, that this antibody accumulates at the hyphae tip. So candidalysin is produced at the hyphae tip. And if you look into more detail, this antibody was produced by QVQ, with the company QVQ, and a group of Sergio Greenstein, Westman and Michel Maxon, they could show that of these invading hyphae, that there's actually an accumulation of this factor here, which is an indicator of membrane repair, epithelial membrane repair. So this fits very nicely to our hypothesis that there's damage here at the tip caused by candidalysin. So next, translocation of Candida albicans through intestinal epithelial cells. Of course, back to the principle of translocation again. Candida albicans cells, hyphae, invade through the epithelial layers into the bloodstream, and we proposed that candidalysin might be involved in this. Therefore, we used an assay which is based on a transwell with 3 different readouts. Okay, just check the chats. It's not important. Right, 3 different readouts. There's first is the epithelial barrier integrity, which is measured by trans epithelial electrical resistance, TER. The second readout is release of lactate dehydrogenase, LDH, and the third readout is the translocation per se by counting colony forming units. And this transfer assay consists of intestinal epithelial cells, including mucus producing cells, to be as closely as possible to the real in vivo situation. Okay, again, checking first adhesion, invasion, and hyphae formation. That's all the same, mutant versus the wild type, and invasion is the same, hyphae formation is the same. You can clearly see here the EC1 mutant nicely forms hyphae similar to the wild type. However, if you look at the three readouts for translocation, so the epithelial integrity is much less destroyed in the EC1 mutant as compared to the wild type and the revertant. There is strongly reduced epithelial damage in the EC1 mutant, and importantly, translocation is also reduced. That means candidalysin-induced epithelial damage mediates translocation. So the summary of this subproject is shown here. You do have damage of candidalysin, which is permitting transcellular translocation through this damaged epithelial cell. You don't have apoptosis, so we can exclude apoptosis, but you have, to a minor extent, probably paracellular invasion, translocation, and maybe even transcellular invasion without damage. So basically, we could show that candidalysin is a classical virulence factor. A classical virulence factor is a microbe A with the gene A, and that's producing virulence factor A, and that's causing damage. This is the definition of a classical virulence factor. So the question then was, is the host helplessly exposed to aggressive Candida albicans hyphae? Investigated Julian Nacklick's team in London, and they published in this publication here in Cell Host Microbes the danger response pathway, showing that a certain threshold of hyphae or the damage caused by these hyphae are recognized by the host, and there are key factors like MAP kinase like P38, which is activating C-phos, and this in turn causes the production of cytokines, inflammatory mediators, and that causes the attraction of neutrophils, mostly neutrophils, but other phagocytes, but mostly neutrophils. And that basically means, but the hyphae moiety and the factor which causes damage was unknown until we find out that this is actually candidalysin, which is responsible for the induction of this pathway. So that means, with other words, that candidalysin can induce a protective immune response. So next, Candida albicans interaction with macrophages. For this, I have a video here. If you just look at this macrophage here, then it's phagocytosis, these yeast cells, and it is damaged. Maybe I'll show that again. So yeast cells here, the macrophage, phagocytosing these yeast cells, then it almost explodes, and that is because of hyphae formation. So Candida albicans can escape from macrophages and can kill its host via production of hyphae, and there are three proposed mechanisms. There's some work on this by different groups. It's from Anna Traven, Leah Cohen, Slavina Vilkova, Mike Lorenz, Melanie Wellington, and Damien Cruzan. And they, based on these publications, there are three proposed mechanisms: pyroptosis, physical forces, and glucose consumption. So of course we wanted to know whether candidalysin dependent escape or killing of macrophages. And again, we used the mutant and showed that the phagocytosis rate of the mutant versus The wild type is similar, the hyphen length is similar, and even the hyphen escape is similar. However, there's clearly significantly reduced damage of the EC1 mutant. And this damage is independent of caspase 1, ASC, and NLRP3, and also
I'm afraid we seem to have lost Bernie for a moment. We'll try and contact him and get him to rejoin.
Hopefully it's just a brief connection error.
Yeah. Okay, this is the perils of having an IT experience. I can't remember ever hearing Bernie stumble, being so quiet in front of a podium over the many years we've had his talks. I think what we're thinking about doing now is we'll contact Bernie, and he's got about probably 8 minutes left in his talk, but I think I'm going to hand over now to my co-host, who's Jay Kolls. And Jay will introduce Geraldine Butler, and we'll hear Geraldine's presentation, and that will give us enough time to try and fix that bit of IT and get you the punchline of Bernie's talk at the end of this. So I'm going to pass over to Jay now.
Great, thanks. It's my pleasure to introduce Dr. Geraldine Butler. She's professor of genetics in the School of Biomolecular and Biomedical Sciences and principal investigator at the Conway Institute at the University College Dublin. She obtained her PhD work, or PhD from Trinity College in Dublin, and did a postdoctoral fellowship at the University of Michigan. And her lab is used a combination of next generation sequencing techniques as well as comparative genomics and gene editing approaches to study evolution and virulence in Candida species. Her talk, Dr. Butler's talk, is going to be ongoing hybridization in Candida species. We're going to, we just introduced Geraldine.
Somewhere around the NLRP3.
Okay, I got a connection again.
What do you think, Neil? Should we?
Bernie,
Yes.
sorry, we were just about to introduce Geraldine because we weren't sure how you'd come back. I'm going to suggest we sort of roll with this. If Bernie, if you could quickly come to an end of that, and perhaps, as you'd suggested before, turn off your camera to increase the bandwidth. And we'll try and get to the end of that. Would that be all right? And sorry, Geraldine and Jay, but we'll just quickly get to the end of that.
I think that's
Bernie, we've got a few minutes to make up to keep everything on time.
Okay, so from my time, I have about 5 minutes left.
That's fine.
Okay, I've been. Okay, I hope you can see this again.
Yes, we can, Bernie.
Okay. Right. So I think it was stopped here. So synthetic peptide of E3 can actually cause inflammasome activation, and that's shown here. So we actually have dual functions of candidalysin. We have both. We do have candidalysin mediated inflammasome activation, but also candidalysin mediated lysis and host cell death. And this is summarizing what we have observed so far with in terms of the dual function of candidalysin. So we do have these dual function for macrophage during the interaction with macrophages. Sarah Gaffin's team actually showed that candidalysin can produce a protective response. Rebecca Drummond could show that there is actually a protective effect of microglia, which promote antifungal immunity via IL-1 beta, and that's also induced by candidalysin. Then it is a classical virulence factor, and it can mediate translocation. So with other words, we have the situation that we have the classical virulence factor here causing damage, but then the host sometimes is able to recognize the virulence factor or to sense the virulence factor, and then in response has an immune response that can actually kill the microbe. So we have both, and this is a consequence of co-evolution. The fungus has invented a toxin, and the host has learned to recognize this toxin and to respond and to actually kill. So we have the virulence factor and the avirulence factor, and both is candidalysin can function as both. So this would be the situation here. We have the commensal situation where we have a balance, then we have disease when there's damage, but then we have a proper immune response, clearance and commensalism. However, that depends on the tissue and on the type of infection, because, for example, in oral epithelial tissue, the candidalysin causes attraction of neutrophils, and that causes immune pathology, and that's detrimental. In this case, it's a virulence factor again. So to summarize my talk, we do have the commensal situation, we have commensal factors, and they have EC1 as a virulence factor, but on the other hand, we can also have virulence function of EC1, so we have dual function of EC1, and there are other examples of virulence factors, which I have not talked about today. So I would like to finish with this statement here: nothing in biology makes sense except in the light of evolution. And I would like to thank many people, in particular my department of microbial pathogenicity mechanisms, in particular the EC1 team, but also Julian Nacklick's team in London. I would like to stress that we are doing all candidalysin related work together. And there are several other global partners, global EC1 team, which are shown here, further collaboration partners shown here, and finally funding, the DFG, the BMBF, the EU, and the Wellcome Trust and the Jena Graduate Schools. And with this, I'm finished, and I apologize for this breakdown here of my connection, and I would be ready for questions.
Thank you very much, Bernie. Apologize that the bandwidth let you down, but it was brilliantly recovered. As you know, we are taking the questions for both speakers at the end. So I'll now pass on to my co-host Jay to introduce Geraldine Butler.
Yeah, great, thanks, Neil. Yeah, so it's my pleasure to reintroduce Dr. Geraldine Butler, Professor of Genetics in the School of Biomolecular and Biomedical Sciences and Principal Investigator at the Conway Institute at University College of Dublin. She's made seminal contributions to understanding virulence in Candida species using combinations of next generation sequencing, comparative genomics, and gene editing. She's published over 100 papers, and today's talk is entitled Ongoing Hybridization in Candida Species. Thank you, Geraldine.
Okay, thanks very much, and thanks for the opportunity here. Hopefully all my screens are going to operate properly. So what I want to tell you about today is a little story that we've been working on for several years, looking at the definition of species in Candida, and particularly the role of hybridization. So I'm going to start with a few questions here, which I may or may not answer, but hopefully I'll give you some things to think about while addressing these. So I want to ask what we mean by a Candida species and what we mean by a Candida hybrid, or I suppose to be exact, what do I mean by a Candida species and a Candida hybrid? And then just think a little about what causes so many pathogenic Candida species to be asexual, what perhaps is the underlying biological reason. And do asexual Candida species tend to be pathogenic? And then finally, the question is whether hybridization in Candida is associated with virulence. Okay, so first, what do we mean by a species? So as I tell my undergraduates, Wikipedia is always a useful source, but perhaps not the only source. And here it's not really very useful at all. A species is a basic unit of classification. Yes, it is, but it doesn't tell us very much. And then our standard sort of undergraduate definition of a species as a group of living organisms capable of exchanging genes or interbreeding. And this is particularly useless when we're talking about Candida, because the term Candida means, by definition, asexual or incapable of mating, or at least incapable of going through a full mating and meiotic cycle. So what we tend to use, at least what I tend to use, and what a lot of comparative genomics, people who study comparative genomics tend to use, is a more molecular biological definition. So here is one taken from definition in bacteria, where species are ones in which the 16S ribosomal RNA gene sequences are more similar than 98.7%. Doesn't tell us anything about the biology of the organism, but it is kind of a useful definition. So if they're more than 99% identical, the isolates belong to the same species. If they're less than that, then they may represent different species. And just for comparison, the difference in Saccharomyces strains is just over 1%. So isolates that we call Saccharomyces tend to be about 99% similar. However, if we look at more closely related species, or species that are closely related to Cerevisiae, like Saccharomyces paradoxus and Saccharomyces quadriceviae, the genomic difference can be as high as 4.6%. So in fungal genomics, we're not really quite clear what defines a species, but we tend to use a sort of a rule of thumb where anything that's less than 4 to 5% different, we tend to think of the same species. And if they're more than 5%, or the genomic differences are more than 4 to 5%, then we tend to think of them as different species. So my next question then is, what is a hybrid? So presumably we all know what a hybrid is. And from here, the Wikipedia definition is actually very useful, because it says it's the offspring combining the qualities of two organisms of different breeds, varieties, species, or genera. So it's all very general here as to what exactly are combining together to make this hybrid species. Unfortunately, it then goes on to say through sexual reproduction. And again, we have a question of how much sexual reproduction is involved. But what we're looking at here are some classic hybrids: a mule or a hinny. The names depend on which is the father and which is the mother. A liger, Durham wheat here, and a cross between a goat and a sheep, which is called a shoat or a geep. Or if we're speaking Chinese, they probably all have exactly the same name. But what these things have in common from a genomic level is that they're highly heterozygous. Because what they have, the mule here has one haplotype, one copy from the horse, and one haplotype from the donkey. So the two alleles are very, very different. They're very distinct. So they're very high levels of heterozygosity in these organisms here. So we can tell, if we knew nothing about a mule and we just had its genome sequence, we could tell very clearly that it was very likely to be a hybrid organism just by looking at the level of heterozygosity. Now, unfortunately, this high level of heterozygosity doesn't always survive. And if we can imagine a mule here lived a little bit more like yeast, and it could divide by budding or it could divide by mitosis, then over time, you would start to lose some of that heterozygosity. And that's a feature, a well-known feature, that you get extended lengths of loss of homozygosity, where the heterozygosity regions have reduced. And that can make it a little bit more difficult to look at the genome of an organism and then decide whether it's a hybrid or not. So when we're looking at organisms like yeast, we tend to use methods like this. And this is a method that I think Toni Gabaldon in Barcelona has applied in a particularly nice way. And what he has realized is that what we tend to see over time are loss of homozygosity in tracts, in shared regions. And that's because LOH tends to occur during, as a gene conversion event. So what we're looking at here in this diagram is a piece of a genome from a non-hybrid organism compared to the reference genome from that organism. And what we're plotting along here are single nucleotide polymorphisms, or SNPs. So every time you see a colored bar here, that indicates there's a difference between this isolate and the reference genome for that particular organism. In this particular example, there are very few SNPs, and they're spread all over this part of the genome. In this one, we see a different pattern. We see places where they're very heterozygous, here and here and here. You see two colors there. It tells you both alleles in the isolate are different to the reference genome. But we also see stretches where heterozygosity has lost, so stretches of LOH. And what Toni has done is try to represent this in a kind of easy to see way. And what he did is he broke the genomes down into 27 mers, short regions of 27 bases. And then he looked at the proportion, each individual 27-mer, and the number of times, the sort of coverage we see it in the genome sequence. So if you look at this plot, for example, this is the plot that you get from a non-hybrid isolate. It's essentially a normal distribution of all the possible 27 mers, and these are the different 27 mers that we see. However, if you look in a hybrid organism, you tend to see two peaks like this. And this peak has about half the coverage compared to this one here. And what this peak is representing is 27 mers that are present in two non-identical copies. So we're getting half the coverage of each compared to a 27-mer, which is present here where both copies are the same. And what I should have pointed out is that I'm talking here about diploid genomes. So we're going to be talking a lot about diploid yeast. So you see two peaks like this, that immediately suggests it's a hybrid. And this peak is measuring the heterozygosity, and this one is measuring the homozygosity. And the little black peak here is telling you reads that are not present in the reference genome. So again, this is what we would expect, because reference genomes tend to be collapsed haploid genomes. So they represent just one haplotype of an organism. And what we're seeing here is that there are a lot of differences between the isolate and the collapsed haplotypes. So all you have to really take from this slide is that if you see two peaks in an analysis like this, it's probably a hybrid. And if you see one peak, it's probably not a hybrid. So then to look at the species that I'm particularly interested in. So we talk about Candida species, but we need to think a little bit about what exactly we mean by Candida species. So this is a tree here of some of the budding yeasts, and I'm using trees throughout the talk that come from Chris Hittinger and Antonis Rokas. This is one of their earlier ones. And what this is showing here, this yellow group, is where we find the Candida species. So sometimes called the Baryomycetaceae or the Mechnicoviaceae, but more commonly, particularly recently, they're known as the CTG SER1 clade, because CTG encodes serine in this clade. We now have to call it SER1 because we know there are other clades which have evolved independently. So this is a look at the CTG SER1 clade. And in fact, many of the pathogenic Candida species are in the bottom here in what's known as the Lodoromyces clade. And what these ones tend to have in common is that they're diploid genomes, and they're mostly, if not entirely, asexual in that they don't have a proper meiosis. Just for indication, Candida auris, the new pathogen, is quite distant. It's way up here in the tree. It belongs to the same clade, but it is quite a distant relative to all of these down here. So one question is, are these diploid asexual yeast hybrids? And Toni has answered that, and the answer is basically both yes and no. So if we look at this Lodderomyces clade here, and we look at Candida albicans, what he has shown very recently is that Candida albicans is in fact a hybrid. It's an ancient hybrid. It's got two peaks here. If you remember, the first peak was the heterozygous sites, and there are very few heterozygous sites. So that tells us that most of them have been lost, which tells us that the hybridization that gave rise to Candida albicans is very ancient. It probably occurred once a very, very long time ago, and all Candida albicans isolates have descended from that. If we look at Candida orthopsilosis, which is here, and metasilosis, which is not in the tree but is very close to it, we see that these both have two peaks, and they're much more heterozygous. The peak here is much higher than Candida albicans. So this is from our work, and we have shown that hybridization in Candida orthosilosis has occurred at least 4 times. The data at the moment suggests that in meta it's happened once, but we question that from some recent data. But it does show you that the hybridization is a more recent event than has happened in Candida albicans. However, if we look at Candida parasilosis here, we get 1 peak, so not a hybrid. And when Toni looked at Candida tropicalis in the sequence genome, we saw 1 peak. So in the last few years, we've been engaged in a population genomic study of Candida tropicalis. And just a little bit of introduction about this species. It's a very common commensal associated particularly with blood cancers, isolated from clinical and environmental sources, and it has a diploid genome. What we did was we collected isolates from many different sources. So most of them are clinical. Most of them came from either the CDC in the USA, in St. James's Hospital in Dublin, or from Spain, from Oscar Zaragoza in Spain. And then we collected some environmental isolates. One of the interesting things about Candida parasilosis is that it's very commonly isolated from the environment. And in particular, it's isolated from soil. So we have some isolates from Chris Hittinger, which came predominantly from Wisconsin. And then we have some isolates from Ireland, and they came predominantly from an undergraduate class. So you see me looking a little bit bored there in the class, but actually usually I enjoy this quite a lot. And this is a class where they go out and collect soil from various places across Ireland, and they try and identify fungi. They've identified new species, but they've also come up with a few Candida parasilosis isolates, Candida tropicalis isolates. And these are some primary school children who've helped us do this. So when we constructed a phylogenetic tree of the 78 Candida tropicalis isolates, what we could see is that most of them cluster down the bottom here, and I'm going to tell you a little bit about this. Could I just ask the administrator, I forgot what time I started at, so if you could just tell me when I did, that would be a help. Okay, so these all cluster together, and then we have these 6 isolates out here that are quite different. And these 6 isolates, interestingly, 4 of them came from soil samples in Ireland. So these are the ones that are colored in red. So undergraduates can be useful. They're sometimes useful. And in particular, these 2 isolates here, which came from a student that didn't even make it out of the, basically had forgotten to bring any soil samples with them. And so managed to just step outside the building and collect some soil from right outside the science building in UCD. And they came up with these 2 novel isolates of Candida parasilosis. Candida tropicalis, sorry. We also identified some Candida parasilosis, but these particular ones are Candida tropicalis. So we're looking at these isolates, and we think that they are very, very different to other isolates of tropicalis, which suggests that perhaps they're hybrids. But I'm going to ignore these for a few minutes, just to point out 4 from Ireland, 1 from the US from soil, and then 1 clinical isolate that came from Spain. But if I just look at the rest of these here. Okay, so these are the group that were closely related to each other. And what we see here is that they form clades. The ones that are colored are supported by other methods of looking at clades. And we do see some geographical connections, like these are all American, these are all American, but then most of our isolates are American anyway, most of them coming from the CDC. And there are clades like this, where you see groups that come from Spain and the USA, and Spain and the USA and Ireland in here. So we don't really have an awful lot of evidence for geographical distribution. The environmental isolates are all in this part of the tree, but again, we have so few of them that I'm not really sure whether this means anything or not. So to go back to these 6 unusual isolates. So if we look at the level of heterozygosity in all of the 78 strains as shown down here, you can see 6 of them stand out. And these are our 6 unusual isolates. And these stand out because they're very highly heterozygous. There are huge numbers of different alleles, different SNPs in these. So these isolates down here have about 1 base in 2,500 is a heterozygous SNP, and these ones go up as high as 1 in 25. So we think that these are very likely to be hybrids, so these pretty definitely hybrids. So if we use Toni's analysis to look at them, 2 of them, our coverage wasn't good enough to get these plots, but what we see are in all these 4 unusual isolates, they've got very high heterozygosity, and in fact, very low levels of homozygosity. So these Candida tropicalis isolates are mules in biological terms. They're very recent hybrids, so the hybridization has occurred very, very recently. This is another way of looking at where the parents might have come from. So what we've done here is we've tried to divide the haplotypes to the 2 different parents. And these reads here in blue are very, very similar to the reference genome. They're like 99% similar to the reference genome, whereas these ones up here are only 95% similar. So this, I think, represents 1 parent, and this represents a 2nd parent. And the 2 parents are somewhere between 4 to 5% different to each other. If we look at all 6 isolates, we see the same pattern, except we actually see 2 different sets of parents. So they all share 1 parent in common, this blue one, which is very similar to our Candida tropicalis reference genome. And then these 4 have a different parent, which we call parent B or haplotype B. And these 2 have a different parent, which we call haplotype C or parent C. And interestingly, our undergraduates who didn't go too far to collect their samples managed to collect 2 different types of hybrids. So they managed to collect these AC hybrids and also the AB hybrids. If we look at the mating types, we get more evidence that these hybrids resulted from mating between related individuals. So in the AB hybrids, the MAT-A, so just to remind you that Candida species have 2 different mating types, a mating type A, mating type like locus A, and a mating type like locus alpha. And in the AB hybrids, the A haplotype came from a reference type isolate, and the alpha from a different parent. And in the AC hybrids, the alpha came from the reference-like isolate and the A from the other isolate. So these are originated from different hybridizations or different mating type events. Okay, I wanted to go back very briefly just to look at some attempts that we've made to try and associate genotype and phenotype in these isolates. And we've done this with the non-hybrid isolates because the hybrids are just too heterozygous. It's impossible to associate particular SNPs with particular phenotypes. So what we did is we used pinning and manual analysis to look for growth defects in various isolates, like we've shown here, over 60 to 80 different conditions. And we used a very simple definition where we identified either a defect was present as 1 or a defect was absent as 0. And then we identified polymorphisms, which I could spend an entire hour talking about how you find these and how you assure them and how you could determine that they're real. But we essentially looked for ones that we thought were likely to be related to phenotypes, so ones that were likely to affect protein coding genes. And what we did here then is we looked for an association between the presence of a variant and the presence of a phenotype, and we used a simple cosine similarity to measure that. And we did find several variants. And I'm just going to look very quickly at one of them. And this is one that was an isolate that was unable to use valine and isoleucine as a sole nitrogen source. So it grows fine in ammonium, it grows fine in glutamine, but it can't grow. The only source of nitrogen is valine and isoleucine. This isolate still had about 40 polymorphisms in it that could have been associated with this phenotype. So we still had to carry out some standard biology. And we looked to see what kind of variation or what kind of mutation might be associated with this. And what I'm showing you on this side is the pathway where nitrogen is used from amino acids, and particularly focusing on this one where valine, leucine, and isoleucine enter the pathway. And we found that one of the genes in this pathway up here, so this is showing you how you get down the fusyl acid pathway. And we found that one of the genes, BAT1 in Saccharomyces cerevisiae, or BAT21 in Candida albicans, had a particular mutation, an early frame shift mutation. So I'm going to skip this slide and just tell you that we used CRISPR to introduce that specific mutation into 3 other unrelated isolates of Candida tropicalis. And hopefully what you can see is that this editing made the strains unable to grow on valine or isoleucine. So what we've managed to do here is a genotype-phenotype correlation, and we managed to recapitulate the phenotype using CRISPR editing. Okay, but I want to go back to my questions here and say, what do we mean by a Candida species and a Candida hybrid? I think here I'm going to use a genetic definition of species. So based on a genomic similarity, somewhere between 1 to 4% difference, we say is the same species. Anything over that, we say is a different species. So our hybrids here are hybrids between 2 parents that vary by about 4.5%. So a question then is that does Candida tropicalis break the rule when we think of what a Candida species is? And the answer is it probably does, but only in a very specific and limited way. So that's all right then. So our next question is, what causes pathogenic Candida species to be asexual? Is it because they hybridize? And is this hybridization associated with virulence? And our answer for tropicalis is probably not, because the hybrids that we found were actually environmental isolates, much more enriched environmental isolates. And it's not clear still whether asexual Candida species are more likely to be pathogenic. Because if we look at this bigger tree now that represents more of the CTG's CER1 clade, what I'm highlighting along here are Candida species that you probably never heard of. So possibly they're not pathogenic, possibly they're not asexual, the names may not actually be correct, or possibly like Candida auris, which is sitting down here, we're going to hear about them soon. So maybe they have the potential to be pathogenic and they just haven't had the correct environmental conditions yet. Okay, so I want to just finish by thanking the people that did the work here. So it's done mostly by 2 PhD students, João Pacheco and Quive O'Brien, who unfortunately did not have the option to dress up in nice gowns when they graduated this year, supported by postdoc Lisa Lombardi, and most of the funding from Science Foundation Ireland and the Wellcome Trust. And thank you.
Great, thanks, Geraldine. So I guess now we'll open up for questions, right, Neil?
That's right. So I think what we've decided, and Geraldine's brought us beautifully sort of back onto time. We've still got about 20 minutes, but we do have quite a lot of questions. So what I thought we'd do is we'd divide them up between, Jay will address the ones for, or ask the ones for Geraldine, and I will for Bernie. And I'll ask a few questions and then pass the buck over to Jay and then backwards, and we'll see how we get along. So Bernie, you've got quite a lot of interest in the infection pocket and where you get the expression of candidalysin. And one of the first questions that came in was from Johannes Westman, and he wanted to know whether phosphatidylserine, which is found in the inner leaflet of host membranes, do you know if that is required for the damage which is taking place? So it's to do with the docking and the mixing of the toxin with the membrane, the host membrane.
So which question is this?
Excuse me, Bernie, this is a question asked at 4:21 by Johannes Westman.
The role of phosphatidylserine.
Yeah,
right. Okay. So yeah, basically this we really don't know. So we discussed the composition of the membrane, and we had earlier data which showed that whether the membrane contains ergosterol or cholesterol plays a role, and that ergosterol containing membrane is better protected as compared to an ergosterol containing membrane, which would suggest that the fungal membrane is susceptible, but not the host membrane is susceptible, but not the fungal one. But phosphatidylserine, I don't know exactly what the function is there.
Okay, Bernie, if I just move on then there's intrigued questions about the other peptides which are generated when you have the proteolysis of ECE1. And do you know what happens to the other peptides? Do they locate in the same place? This is asked.
Okay, the other peptides, this is actually the key topic we are working on at the moment. So we do have a number of hypotheses, and we have preliminary data. One idea is that these are actually required for fungal biology itself, for the processing, for the secretion in the Golgi and the ER. It might also be that the other peptides act as antitoxins, so that would be internal protection, that the other peptides protect the fungal membrane within the ER or the Golgi by interacting with candidalysin. We do have preliminary data which suggest that there's definitely a role for the fungal biology, because if we delete certain peptides or if we overexpress candidalysin, then we do have reduced hyphae formation, which is reduced growth. And this could be due to unfolded not proper folding, not proper processing, or not proper sequencing. That seems to be likely. There is also the possibility that the other peptides modulate candidalysin functions. They might prevent integration into the candida membrane also from outside, not also only in the ER and Golgi. It might, the other peptides might modulate pore size and structure. And finally, my favorite hypothesis is that the other peptides might actually act as effector peptides, that candidalysin is causing damage and the other peptides actually released into the host cells and do have a function in the host cell. We don't have evidence for this at the moment, but we do have evidence or preliminary data that the other peptides, when added extracellularly, they do cause, for example, expression of cytokines. So these are the three possible functions, and there might be a mix of functions, but the biology, the function for the biology of the fungus, that's clear. We do have clear evidence for this.
Thanks, Bernie. So you partially answered another question which came up, which is how Candida protects itself from candidalysin. At least you presented some hypotheses there.
Yep.
Somebody did ask, by the way, why, if the mutant of ECE1 makes normal hyphae in terms of, it's able to do so, why was it called ECE1? And I think my memory of that was that the gene is expressed in proportion to the extent of cell elongation. So I think it was named after its transcriptional profile. Is that right?
It was one of Bill Fondi at that time was searching for genes which are expressed in association with hyphae. It is expressed at all time when hyphae are produced, and it's one of the most strongly expressed genes during hyphae formation. And it was actually used by many people as an indicator of hyphae formation. It's always expressed when hyphae are produced. So far, we know that.
Okay. I propose actually what we do is we now pass the baton, and we can pass it back, and we have a few questions for Geraldine and back and forwards and see how we get along.
Great.
Okay.
So Geraldine, there's a couple questions around C. auris. Is C. auris a hybrid, and what is the proximity or hybridization ratio in that?
Right. So C. auris is a haploid. So it can't be a hybrid in the way that I've been discussing. So, but the question is, what is the diversity among isolates that we call C. auris? And there have been several papers recently, but I don't know off the top of my head what that diversity is, but it's nowhere near 4%. They're much more similar.
Great. And then Joe Heitman asked, with these environmental isolates of C. tropicalis, do you think that these novel hybrids are descending from a tetrapolar parasexual intermediate?
So my guess is they are, yeah. So I guess they're mating in the way that we understand tropicalis mates, which is two diploids making a tetraploid and then degenerating back to a diploid. So they do look very similar, but I don't know that.
Okay, great. And then in terms of regulating gene expression, how do the alleles regulate gene expression? Is there any evidence of like allelic exclusion or preference of one allele over the other?
Yeah, so that's a good question. I can't answer that yet. Most of these hybrids we've only identified in the last year or so, and we haven't done any transcriptional studies, but I would like to do that, and that's one thing I really would like to look at.
Great. Julia Cronin asked, is there any other evidence other than hybridization that mating occurs in Candida species, and what can cause mating to occur?
So what can cause mating to occur in these diploid isolates?
Yeah.
So that the ones that are best studied are albicans, dubliniensis, tropicalis, and so there are two mating types. You do get mating between A cells and alpha cells. If they have, one is 2 A loci, the other is 2 alpha, then they make a tetraploid, and then they go through something that isn't quite meiosis, a concerted chromosome loss, and they end up back as diploids again. And Richard Bennett in particular has done a lot of work studying how that happens and how that's regulated.
Great. And Ilya asked if moving forward, what are we going to call a strain in the fungal and particularly Candida world?
Yeah, so that's a good question. So I think what we have to accept is that our definition of species is not very clear, especially when we look at Candida, and the more that we isolate, the more that we sequence, the more diversity we realize is there. So I did see someone asking me, how do I know these are tropicalis? And I should have made that a little clearer when I showed the tree, that if I put other species into that tree, they're very definitely tropicalis. If you sequence the ribosomal DNA locus, which is what you'd normally do in a lab, you'd say this is tropicalis. So they're much closer to tropicalis than they are to anything else. And they're not a different species because one of the parents is the same. So I think we have to start thinking about Candida in a much wider range. And now that Toni has shown that Candida albicans is an ancient hybrid, there is a possibility that there are other hybrids, Candida albicans as well, that we just haven't yet identified. So we may have to expand our understanding of what we think of as a species.
Great. All right, let's move back over to Neil.
Okay, Bernie, first question, Sifu, what do you think about the origin of candidalysin? Where do you think it comes from? How wide is it shared? That sort of question. I think that's what we're getting at there. The evolutionary origin.
Yeah, that is a very interesting question. So, of course, we don't know exactly where it comes from, but this is the same discussion as the discussion in other pathogenic microbes which also produce toxins, which are toxic for host cells. And the discussion here goes along the time where the host was not there, and that these toxins might play a role in competition with other microbes. And we have the same hypothesis that potentially candidalysin was produced for competition with other microbes in a microbial community. And we do have preliminary data that candidalysin in fact acts against bacteria. So potentially this is the evolutionary origin that originally has been evolved to for competition with other microbes.
That's a very interesting point. So this microbial warfare works in both directions, as we're finding out. Okay,
Yep.
so Carol Kumamoto is asking, so you started off in your presentation showing all the different pathways by which Candida gets in. And does ECE1 participate in any of these other pathways, the paracellular pathway, the transcellular pathways, for example? Any role there?
Also a very good question. From our data, we would propose that it does not play a role in the other translocation routes, because the ECE1 mutant, and this is actually why we propose that there are additional routes. Now, the ECE1 mutant is not completely inhibited in translocation. It still can translocate to the wild type about 25% in our hands. So that's why we propose that the remaining potential is actually paracellular, remaining translocation route is actually paracellular. It might be that if you discuss the difference between these and whether that has an influence on the hyphae which actually translocate, it might be that these hyphae, when they are invading and penetrating through epithelial cells, that the hyphae properties will change during this procedure. And that might be different from translocations through damaged epithelial cells and the paracellular route. And we actually in investigating this, whether the translocation per se might change hyphae properties.
Very interesting. Here's an interesting question from Julia Palmucci, that noting that melittin was found to be a selective target for HER2-enriched breast cancer cells. Any information about receptor selectivity for candidalysin?
Yeah, also very good question. This is actually Julian's territory. So he's working on the receptors, and the situation at the moment looks like there is no direct receptor, but what is sensed is actually the damage caused by candidalysin. So basically, the receptor is unknown, but the damage causes the release of alamines and AMPs. And Julian has shown in 2 publications from his group that EGFR seems to play a role as a receptor via this, but a direct binding, it seems not that likely at the moment.
Okay, I'm going to take one more question, and I'm going to apologize at the same time. There's just such great questions here, so I do apologize if we don't have time to get to yours, but we can keep a record of all the questions, of course, and Bernie can take them away and perhaps get back to you. But Mark Swidergal's got an interesting one, if you see that there, about noticing that the mutant, the EC1 mutant, doesn't persist in the oral cavity. So damage, therefore, we deduce is not a driver of commensalism. So what factors do you think contribute to commensalism?
I wouldn't say that it is that simple. So we know that our favorite lab strain, SC5314, is not a very good colonizer in the oral cavity. So it's causing damage and it's causing inflammation, and then there's clearance. I would propose, and SC5314 is actually an exception in if you compare this with other clinical strains, it is pretty aggressive. And this means it's a pretty well hyphae producer, and at the same time, a pretty well EC1 producer. So I would propose that you need a combination of things which causes damage. And that's including hyphae formation per se and EC1 production, but also potentially other activities associated with hyphae formation, proteases, sub4 to 6 are also hyphal associated. And it's also should also be stressed that good colonizers, clinical isolates which are good colonizers, are actually non-hyphal producers. So the secret here seems to be hyphal formation versus non-hyphal formation.
Thanks, Bernie. So I'll pass back the baton to Jay to pick up with some Geraldine questions.
Great. Thanks, Neil. So Geraldine, Professor Alistair Brown asked, you suggested albicans as an ancient hybrid. Is it possible to implicate contemporary species as derivatives of the two parents involved in an ancient hybridization event?
Right, so maybe I wasn't totally clear that the albicans work is from Toni Gabaldon. So he's got a very nice recent paper. I have a theory that Candida africana might be one of them, but he doesn't think that it is. So right now, no, we don't know if the parents are still around or not.
Okay, and then actually, you answered a technical question by text, but I thought maybe just to clarify for the audience too, is the hybridization, the workhorse of what we call a hybrid is based on NGS genomic sequencing. Is that the definitive assay?
So yeah, the definition of a hybrid depends on knowing the heterozygosity level, and that needs a whole genome sequence.
Yep.
Yeah. Otherwise, it doesn't look. Otherwise, it would just look like Candida tropicalis.
Yep. And then a question from Constantine Martinez at San Vicente. There seems to be many Candida species that hybridize. Is there any hint in the genomes of these species that tell us, why this is? What? I guess there's an evolution advantage for
The reason we know that there are lots of Candida species that hybridize is because we've sequenced lots of isolates from the individual species. And we've done that because they tend to be associated with pathogenesis. For many other species, we sequence one. So we don't know how common hybridization is across the CTG clade, across budding yeasts, across fungi. Maybe it's a lot more common than we realize. You see it a lot in Saccharomyces. So I don't know if it's something specific to these species, or is it actually a very general phenomenon that we've just been ignoring, or we didn't realize was happening.
And Joe Heitman asks, can you find other parents of the novel tropicalis hybrids in nature? So
yeah, so probably, possibly, I don't know. We'd love to look, yeah, but we don't have them.
Okay. All right, Neil, I'll pass it over to you.
Well, just to keep things on time, I just want to wrap up, but just want to thank both of our speakers, Geraldine and Bernie, for fabulous talks. I'm sure you've been really enjoyed that. There's over 240 of you, by the way, listening into this talk. So again, a wonderful attendance. I also want to thank Jay, my co-host, the organizers of the MycoTalks, and all those people who asked questions. And again, sorry to disappoint some of you that you couldn't get the answers directly during the MycoTalk, but we have a recording of this whole talk as you had at the beginning. And so you'll be able to continue those discussions directly with either Bernie or with Geraldine as you see fit, and they'll probably be very interested in answering some of those questions. So thanks again.
Yeah, just a reminder to register for the October 29th talks, which will be Sarah Gaffin and Alex Brent.
Yeah, the 29th, put it in your diary, make sure you're registered, and we'll look forward to seeing you or having your presence at that talk in a short while. Thank you very much, everybody.
Yeah. Thank you, Geraldine. Thanks.
Okay.
Bye.
Okay.